Then, which of the following multivariate analysis follow interdependence model? What exactly this interdependence model? For example, you having a yield. For example, you having a yield. You having a yield. It is a dependent variable, and you have number of pots, number of tillers, number of seeds per plant. Like this, some trait they are independent variables. Independent variables. For example, if you are analyzing your data, for example, you are doing some analysis, but you are having 1,000 plants and 18 like so many independent traits, and you have to compare among them. What like, but it's a huge data. Data is huge. Means what we will do? We have to reduce. We have to reduce. You have to reduce the dimension of the data. Dimension of the data means you have to make into, you have to like 1,000 traits into 6 groups. It is called as principal components. In this 6 group, by comparing 6 group, you can make some sort of interpretation. But you having 1,000 groups, what you will do? What, how you will compare? Like one is greater, one is greater. But you have to reducing into 6 groups, 6 principal components. Then you can interpret the data. But this is in this we are not taking, okay, if this independent variable you have to take separately, dependent variable you have to take separate. Not like, it is not classifying and comparing the dependent variable and independent variable. That type of model is called as interdependence. Means it is not taking, it's not comparing the dependent variable and independent variable, then it's called as interdependence model. Your PCA is interdependence model, but rest are all dependence model. See, multiple correlation. You are like a multiple correlation. What you will do? Your independent variable will be there. You are correlating with dependent variable. Okay, independent variable is correlated with dependent variable. Here also discriminant function analysis will do same. In canonical correlation also we are doing the same. We are comparing dependent variable to independent variable. Means it dependence model. It's not interdependence model. That's why principal component analysis is the answer. Then, which of the peptide addresses the label target of the proteins to chloroplast? Actually, these are the questions you need to by heart. There is no other way. I will show the answer directly. Actually, transit peptide is especially related to chloroplast, and pre-sequence is related to your mitochondria. Signal peptide is related to endoplasmic reticulum, and localization of pre-sequence is not specific to anything. Like that, we have to remember. This is a basic question. Like when you are reading the book genetics, we are normally avoiding the chapters. We are reading up to translocation, like translation chapter, but we are not going to read post-translation, post-translation modification chapter. Post-translation modification chapter. Please read that chapter, then you will get it. Then what is erucic acid? It's also a beheaded type of question. Like you have to remember MAGA, mono unsaturated 22 carbon fatty acid. And you should know what is erucic acid, how to breed erucic acid, what is the importance. It's a quality chapter. If you have to read quality chapter book, quality chapter from the BD Singh, then you will get it easy. Then it is also very simple. Which strand is, for example, this is DNA. Which strand is go for mRNA? Which strand is going as mRNA? For example, if this is going to mRNA, this will be the your template. It is called as an antisense and non-coding, because It is a complementary to the strand which is complementary to the mRNA, but other strand is exactly look like your mRNA. Like it's called as a coding strand. These are the basic things you are reading from last since PUC. This is a simple thing, but many times you will confuse. But you have to sense and coding strand and anti. It's not there. Sense and coding strand is one and the other same, and template strand and anti-sense strand is one and the other same. You have to remember those things. Then the selection is most and the least effective when the gene frequencies are respectively. For example, selection is best when your allele P and Q is equal. P and Q are equal. Like if P is 0.5 and Q is 0.5, then your selection will be high. Why? Please read the chapter. Please watch my video. Selection components. It's there in the YouTube. Then you will get it. Why it is. Then selection is P because see Q is 0.01 and another allele is 0.99. Means here one of the allele is fixed in the population. What we will do? If allele is fixed in the population, what we will going to select? Okay. That's why if allele frequency is P is equal to 5, your selection opportunity is more. Then you select better plant. That's why when Q is 0.5 and Q is 0.01, selection is high in Q. P is equal to 0.5 and Q is 0.01 least. Please read. Then theoretical melting temperature for the this one, I think there are some formulas like 2a plus t plus 4g plus c. Like one formula is there for melting temperature plus or minus 5. You will get annealing temperature also. But don't use that much overthinking concepts. Just for PCR, what we need? Forward primer plus reverse primer, not plus reverse primer. And if you are setting in a single reaction, means you should have equal melting temperature. Check 31.8, 32.8, not 64.8, 65, not 50.8, 50.8, not. If you have different melting temperature, how will PCR will happen? Like how PCR reaction will going to happen? See, that's why you should have equal melting temperature for both forward and reverse. Then only your PCR will multiplication will happen and you will get a product. Then it's also a very simple question. If you know three things: what is foreground selection, what is background selection, and recombinant selection. What is foreground selection? You are using a marker to select desirable trait. What is background selection? The markers are used to select recurrent parent genome. Recurrent parent genome. Then recombinant selection to avoid a linkage drag. If you know these things more than enough for any exam. To avoid linkage drag, background selection to recurrent parent genome, to recover the recurrent parent genome, and foreground selection for the selection of desirable trait. Okay, then you will solve this question. What exactly it means? See, if a dominant disease resistant R gene linked with a dominant marker, means dominant marker, and small r is linked with small r marker with 300 and the marker showing 300 base pair. Applicant coupling phase, and they are in coupling phase, means dominant with dominant, recessive with recessive. Okay. Recessive with recessive, sorry, dominant with dominant, recessive with recessive. I think you know what is coupling and replenishing phase. Then the marker should background spreading of a form to transfer the gene using what if you want to select this background selection based on the presence of 300 base. No, I told background selection is for recurrent parent recovery. Okay, genome recovery. We have to use an unlinked marker from the donor parent. It is not the option. It is actually uh, not it's not actually, it should be a foreground selection. Foreground selection based on the option, no, it should be presence. Foreground selection based on the presence of 3. This will be the right answer. And again, this will be not background. When you're selecting desirable gene, you should automatically click foreground. Then it should be presence, okay? Because it's a coupling phase. If it is in a repulsion phase, please answer in the comment box. Then the variation in the size of the tRNA is because of variation in the length. It's a direct question. Dearmament extra. Like we know that this is like 5 prime to 3 prime. If there is a variation in this d-arm and variation loop, sorry, d-arm and variation loop, your length of the size of the your tRNA will going to change. It's again a direct question uh, from the paper. Actually, these things are very nicely taught in our classes actually, but I don't think in any other essays these things are not taught well, because we have to always look into the recent papers like crop science, tag, like theoretical and applied genetics paper, plant breeding, and nature publications. You have to read the paper and recent concepts, and which is important. If you read PK Gupta book, these things are available. LR34 gene is the first R gene cloned from the wheat. No, there are other gene as cloned. Actually, I forgotten. And LR34 gene encodes ABC. This is a very good point. Like ABC transporter only. Like every gene having a function. It's an ABC transporter. It's a direct question actually. It is actually taught in our classes, but it is, I don't know. Directly we opt it. But you have to read the recent papers, okay? Like there are, because green revolution happens, why important traits, one is short duration and another is like short duration not like semi dwarfness, and another is rust resistance also. When we are reading, we are only focusing on semi dwarf, but we are not focusing on rust resistance, okay? Because of the rust resistance only, like resistance also is one of the factor of green revolution. Then how it has happened, which are the genes which revolutionize the like rust resistance, it's a very important thing to read, especially blast and blight and related research articles in rice and rust in wheat and leaf curls in sunflower. There are important is late blight in potato. You have to read the biotic stress chapter from BD Singh to get these things, and you have to read the recent articles of this important disease, and it's a mandatory also. You should know these things perfectly if you want to become a good breeder. Then MC Clintock, we all know that she worked on MAGE, and and the pseudodominance is due to deficiency. It's a direct question. Then, if you two inbred lines, a single cross hybrid differing with the k number, like p and parent 1 and parent 2, they are having k number of loci. In case of, for example, if k is equal to 10, if like 10 loci were there, like past a b c d, like 10 loci were there, then in the case of extreme dispersion, all the positive alleles of increasing value on a phenotype percent in the, like there is one formula for the extreme dispersion, like rd is equal to kd minus rd is equal to kd minus 2k dash d kd. I am also writing by seeing actually. Sorry. rd rd is equal to kd minus 2k dash d by kd. Means what exactly these things are, what exactly coefficient dispersion means. If coefficient of dispersion, this RD value is equal to 1, means it's a extreme dispersion. Extreme dispersion. What exactly it means? Extreme dispersion means, for example, if parent 1 is there, parent 2 is there, you have to make an hybrid. If parent 1 having all 10 positive allele, if parent 2 having all 10 negative alleles, negative genes, then in that case, the RD is equal to 1. And another case, you having 5 positive allele and 5 negative allele in one parent, and 5 positive allele and 5 negative allele in another parent. This is called as complete dispersion. Complete dispersion. Then if there is a complete dispersion, then RD is equal to 1. Okay. Sorry, RD is equal to 0 at that time. I think you came to know what is. I will explain again. Sorry, I made a mess here. Messed up everything here. Okay. For example, there are two conditions. One is complete dispersion and one is extreme dispersion. Extreme dispersion. Here also D. In complete dispersion, parent 1 is there, parent 2 is there. Here you having like 10 loci you take. K means any value you can take. 5 positive and 5 negative. In parent 2 having 5 positive and 5 negative. Means it's the case of complete dispersion. Then the RD is equal to, at this case, is 0. Then for extreme dispersion, parent 1 and parent 2 if you take, having all positive 10 alleles in this, all negative 10 alleles in this. Means all positive alleles is concentrated in parent 1, all negative alleles is concentrated in parent 2. Means at this case RD is equal to 1. Then this is called as extreme dispersion. Then then the formula to calculate this, you take KD minus 2K dash D by KD. If you use this formula, you will get. And read these things in the heterosis breeding by Mukherjee book. It's available in IRA bookstore. Please order it and read it. Then why we need these things? Why we need to, when if RD is 0, when RD is 1, what will happen? For example, if there is an extreme dispersion, the chances of heterosis will be less. If there is a complete dispersion, chances of heterosis is more. Like H is equal to dy square. Remember this formula. What is d, what is y, how it is calculated, please remember and read this chapter. Okay. Then again a very interesting question. It's a 4EMS system of producing hybrid wheat. See, when you look into the hybrid wheat, you know, in hybrids in maize is famous and hybrids in rice is famous in China, but nowhere we heard about hybrid wheat. In books only we are there. Okay. But what is hybrid wheat and why it's not famous in the wheat? Because of it's highly self-pollinated crops and there is no male sterility systems available. The effect it's not no male sterility system. No effective male sterility systems are available in the wheat. Not available in the wheat. There is one method like 4EMS system is developed to it's an advanced technology for the developing of hybrid wheat. It's a male sterility system using 4E alien addition line. Alien addition line. It having some color genes. Based on the color of that particular gene, we are designating maintainer line, fertile line, and sterile line. We are identifying fertile line, sterile line based on the color, and the color is coded by the gene, which are gene which having on the chromosome number 4E. We will detail it's published in crop science. That's why reading recent papers, not recent papers, at least important impact factors, 4EMS system. There are different systems of male sterility. If you read this paper, you will get to know, and I will explain. For example, okay, for example, you having blue color. Also, I didn't solve the question. For example, read this paper, you will get the answer. Actually, for example, if there is a complete blue color, then your plant is, what will happen? No sterility. There is, because it having the all the genes. Sorry, the figure is not good. Okay, I will explain. If the, but three things here: if the complete blue color, no sterility; if you're having a partial blue color, somewhat of male sterility, but most of the time male fertility. If there is no blue color and all are white, then there is a complete male sterility. And we are selecting based on the color. I'm not going in detail because it will confuse you. I will explain these systems in my separate classes, hybrid recent advances in hybrid breeding. If you want to watch that video, like if you want to learn that this concept, please subscribe my channel. Which of the following methods can be employed for the production of near isogenic lines? What is near isogenic lines? For example, if the two lines is almost all having a similar genotypic constitution with a light difference. Isogenic line means only single gene difference, but having small difference, like a near like one or two genes difference in the two individuals. The rest all these are same. But how we can create? By backcrossing. Exactly, you can transfer in a single recurrent parent background from different donor parent 1, donor parent 2, donor parent 3. If you cross, you will generate R dash, R2, R3. Like donor parent 1 gene, donor parent 2 gene, donor parent 3. We can create the nils. And as, sorry, I think this is selfing. Yeah, actually, even selfing also we can able to create a near isogenic lines. You can read the background chapter, you will get to know. And mutation, if you create a point mutation in the both the lines, we can create a nil and chromosome substitution. Like it's a ABCD. In chromosome substitution also will create near isogenic lines. Please read the chapter how we can create by duplication and deficiency lines can be used to create nils. Please read the chapter. Then the nature of gain function mutations are always. See, they are asking a gain function, not a loss function. If a gain function means it's almost a dominant in nature. What is gain function mutation? What is gain function mutation and loss function mutation? Means by mutation you are gaining some function. For example, if A to T transfer happened, means the leaf will develop. If loss function means A will going to lose, then if your leaf will going, the leaf formation will not going to happen. Like this, if function is generated by mutation means it's a gain function. If function is lost by mutation is a loss function mutation. Then excess of coupling phase linkage leads to overestimation. See, asking coupling phase means additive gene action. It's a direct question. Please read that. If you want to know more about this, please subscribe my channel. Then this is a very simple question. Again, degeneracy of genetic code. See, CUG codes for N-formal methionine at an initial codon. It's exactly right. But another position is codes for valine. Yes. If you read codon, this codon table, you will get to know. It's a direct question. No need to discuss. And stock 6 is a paternal. No, it's a maternal. What is maternal haploid? What is paternal haploid? Why it is used in the hybrid breeding in the maize? What is haploid inducer lines? What is haploid induction rate? You should know these things. For example, stock 6 is an important haploid inducer line. It's a maternal haploid. If you want to learn these things, please subscribe my channel. Then, paternal haploid induction stock 6 in stock 6 results from mutation in the IG. Okay, actually, it's a, both are wrong. Please check the answer. Triticum spelta, allohexaploid in nature. Exactly. And ABDs, exactly. What is AA? What is AA, BB, GG? Please comment your answer in the comment box. Which of the following location is the most suitable priority undertaking the germplasm? It's a microcenter. For example, you're having an origin of center. You're having a microcenter, you're having maximum variability and diversity. Means if having maximum variability and diversity, automatically what will happen is chances of selecting good ones or identifying a new species, it's a very easy. That's why microcenter is very important. The real value of a seed lot is determined by, please answer in the comment box. Okay, it's a very easy question. Then the 2 ANS translocation was developed by using Agilov tox. No, Agilov's ventricosa. It's a direct paper. Actually, it's picked from a Priyanka Agarwal paper 2021 published from IRA authors. Please read this paper. It's a direct question actually. Then, which of the following methods is a form of extensive germplasm conservation? So, natural park, it's an in situ. Field gene bank is extensive. Gene sanctuary is in situ, and microcenter is in situ. And field gene bank was the answer. And please subscribe my channel for other videos. Like I have discussed only 25 questions, I think. For other 25 questions from part C, for other 50 questions from part B, and GK question 20 questions part A, I will discuss in a separate videos. If you want to, if you like this video, please subscribe, like, share. Thank you.